Okay, so where we left off was on, so we talked about all these different groups of protists. We also, just a couple refreshers, discussed the fact that protists are overall rejects. They tried to be plants and didn't make it. They tried to be animals and didn't make it, or tried to be fungus and didn't make it. And we've talked about a few different groupings and the characteristics of those groups of protists. I also talked about study strategies for this stuff because it's going to be straight memorization, where you might see some overlap in some of these characteristics. A specific protist has this specific set of characteristics that may be similar, but there is some distinguishing characteristic, and you just need to make sure that you're familiar with that. And that's where these chapters kind of become convoluted, because you have bacteria, protists, and fungi, which are all really exciting topics to study that all have very similar yet different or distinguishing characteristics. So a lot of memorization. So where we're picking up are on the heteroconts. The heteroconts have flagella, one of their defining characteristics. There are 4 groups of heteroconts: Oomycota, diatoms, golden, and then brown algae. And we're going to speak on each individually. Beginning with Oomycota. Not Oomycota, but Oo. Oomycota is a fungal-like protist. So it tried to be a fungus and was lacking some defining characteristic, and so as a result, it's a reject. It includes water molds, white rust, and types of mildew. The characteristic here is they are saprophytes. This may be a new term for you, but it's very similar to terms that we've discussed before, like detrivore. What's a detrivore? Dead or decaying organisms. Same thing as a saprophyte. Okay? But what does phyte tell you? It has some type of plant. Like, do you see where we're making that? Like, it's a reject. Saprophyte. Could also live as a parasite. These grow as masses. And it uses the word highfill. Now, you and I have not talked fungus yet. But as soon as we get to fungus, you're going to see this term a lot. Hyphae are the filaments that are used to construct fungi. So when it says something highfill, that tells me this is a fungus-like protist. If I didn't know it was fungus-like, just the characteristics alone, using hyphae filaments, tells me that it's fungal. So you'll see that term show up again. Another term that you will see is the term mycelium. And we'll discuss that, of course, in more detail when we get to fungus, but a characteristic of fungus. Look what other characteristic Oomycota have. They secrete enzymes to digest organic material. Any idea what type of organism also secretes enzymes to digest organic matter? Just a shot in the dark? A fungus. Does anybody know how a fungus eats? So they're heterotrophs. A lot of people mistaken fungi as being autotrophs because a lot of them come from the ground or found on plants. So they think that they're photosynthetic. Fungi are not. Fungi are heterotrophic, and the way that they consume their food is they spit out digestive enzymes out of the top of them, which we'll talk about, their fruiting body. They will secrete enzymes, and it will break down the ground around them or the material around them, and then they will soak up what they've broken down. So it's like extracellular digestion. They're digesting it outside of themselves, and then they absorb it, and it causes them to get bigger. And then they secrete enzymes at a bigger range, break all that stuff down, and then they absorb it, and it continues to do so. So fungi are heterotrophic, but they don't have a mouth. They get their food by breaking down, digesting it extracellularly, and then absorbing it through their hyphae, and we'll talk about hyphae. They do not have roots. People tend to think that they have roots. They don't. Fungi do not have roots. They are not plants. Some examples of Oomycota, kind of like here is mildew on some fruit. Like you see this kind of stuff. All we're doing is classifying it. Water mold, like when you have fish that expire and they're kind of floating, then you pick them up and they have that like sliminess to them. That's the same thing. So it's feeding on, it's a saprotroph, dead or decaying organisms. Just looking up close. Again, when we talk fungi, we'll talk about hyphae, but here's a hyphal filament here. Hyphal filament. Hyphal filament. When I teach them to you, I'm going to say they kind of look like spaghetti strands, because when they are all put together, they look like a whole bunch of individual spaghetti strands. Another example, we have this fungus like Protists living on a plant, but it's turned this area darker colors. Because the chlorophyll is not exposed, guess what it can't do? Carry out photosynthesis, right? So the rate of photosynthesis of this organism is going to continue to decrease until eventually it expires as that protist continues to spread, that mold. The next group, diatoms. So my Coda was the first group of heteroconts. Now we have diatoms. So in diatoms, there's a couple words that are going to come up here. They're going to talk about shells, and then they're going to refer to those shells as tests. So these organisms, and I mentioned it last lecture, but they create their own shell. They secrete it. So it's an organism, and they carry out a metabolic process that secretes a shell. So that it's like they grow their own shell. And when it becomes solidified and it's a solid mass, we call it a test. When we look at these diatoms, and this is what comes up, what we put together as diatomaceous earth, so if you've gotten to that part of the lab, it's pretty cool. They have very unique structures and shells. They don't look the same. Some of them are autotrophic, some of them are heterotrophic. There's a range of diatoms. Some of them are able to asexually reproduce, and some of them sexually reproduce. Now here, I have two descriptions of the shells or tests that these organisms are going to have. And it says here, daughter cells receive different sized shells. Are daughter cells the result of asexual or sexual reproduction? Asexual. Daughter cells are the result of asexual reproduction. Here it says the zygote will secrete a new shell. Sexual or asexual? Sexual. Zygote is an indication of sexual reproduction. That's where sperm and egg have met. So these two distinguishing characteristics are following along with, like if you really read into it, you would tell the difference between sexual and asexual. Well, that's what I'm trying to get you to understand here, is that these are not, all of them have that same characteristic. Okay, so again, it talks about, well, it gives those characteristics. This continues. So here is talking about that shell, and it calls it, they secrete a silicon dioxide test, or a shell. So you see the term test integrated in here. That's that shell structure. That it's called a frustule. This forms diatomaceous earth. They exhibit both lateral or bilateral symmetry and radial symmetry. What is bilateral? Same on right and left side. What is radial symmetry? Like a round, like a pie type thing. So every slice is the same. So of course you can see here bilaterally symmetric, radially symmetric. Where do we find or use this diatomaceous earth, these diatoms? You'll find them like in your pool filters, your fish filters. In types of polishes, we have different degrees of roughness is the word I'm thinking of right now. That's not, it says abrasives, but like where we try to make it coarse to break things down. Water quality testers. And you can also use diatoms to date fossils. So we can use them because they are living organisms. We can look at their molecular clock and tell how old they are. So we can use them if we're studying geology, not geography. Golden algae is called golden algae because it has a golden color. Characteristics. First of all, Christophyta, it forms colonies, so it's colonial form. A colony is not one organism, it's a group of multiple individual organisms. A colony is a group of individual organisms. Okay? So a group living together, and we've already talked about the properties of colonies before. It mentions that they have flagella, which is used for long distance movement. It also has a brown coloring from the pigment called fucoxanthin, defining characteristic of golden algae. Defining characteristic of golden algae. Brown algae has that same pigment, that same brown pigment, fucoxanthin, but it also has chlorophyll A and B, so it tends to have a darker color. This is what makes up our brown algae. Brown algae are usually what we call our large leafy seaweeds. These can grow to 200 meters in height, and I think I've mentioned that before. We found them in a marine habitat. When I say marine, what does that tell you? Salt water, right? And a lot of times, if you're flying over the ocean, sometimes you can see like small dark spots. Dark spots, when you see them, can signify a couple things, but when they're small and within a range, usually those are individual little kelp forests, and you have a bunch of brown algae there. It could also, a dark spot could also say that there's a reef there because it's a little shallow, or that it gets super deep there. But usually when you're flying over, you can kind of see like this forest of kelp, and that is brown algae. We can harvest this kelp, and we use it in our ice creams, our salads. We use it as a stickiness in our candies and foods. So it gives that characteristic. Okay, then next grouping, Cercozoa. Cercozoa are a type of amoeba. So we have a whole nother grouping called amoebas. So I want to make sure we distinguish these. These amoebas have a shell or test. So they're stiff when we talk about Cercozoa. If they fall into this Cercozoa category, they're a form of amoeba, but they're a stiff amoeba, whereas regular amoebas are really slimy and nasty, but we'll get to them next. So Cercozoa are amoebas that have a shell or a test. 3 groupings: Radiolera, Foraminifera, and Chlorarachnophytes. Now, as far as your notes go, we are going to discuss the first 2. The third one is not brought up in these notes. I'm just letting you know that. It is touched on. We talked about this in lab, the chlorophytes and the charophytes, which are next. But there is no notes about this, and so it doesn't go any further other than knowing that it's a grouping of Cercozoa, in case you're wondering that for study purposes. So the radiolarans, it talks about the fact that their test or shell is they have an internal skeleton and it has the components of sediment. So the other one was silicon dioxide. Now you can see a distinguishing characteristic. These are marine heterotrophs, and they use pseudopodal rays. So this is their skeleton, but the amoeba-type organism kind of stays inside of it, and when it wants to eat, it shoots out axopods. So it kind of looks like, this isn't specifically a radiolaran, but it shoots out its nastiness through the holes in its skeleton. So it shoots it out so that it can grab stuff and then it brings it back inside. When we talk about amoebas are a mass of goo, that's what it feels like. So this organism here has a protective outer skeleton to kind of protect that gooiness to it. So that still nasty sliminess is there, but it shoots through and then it's protected by this skeleton. And they're kind of cool looking, again, pretty unique. So I just wanted to show you that picture. I'm going to go back. That's back a slide. So radiolarans and Foraminifera, or also called forams. Same type of thing. Marine heterotrophs. Talks about how they will shoot through those perforations in their shell in order to catch their prey. These are, did I already say that they're heterotrophic? Okay, I thought I did, but then I read back over it. Where will I find these? Their component is limestone. So we talked about radiolarans had like a sedimentary type composition. Here we have limestone. You may also find them in marble. These are symbionts with algae, some of them. What is an algal symbiont? It has a relationship with algae. Understanding or just being able to decipher the term. Symbiont means that it's a relationship between two living organisms. And in this case, it gives you a little bit more information. It says they're algal symbionts, which means that they have a relationship with algae. Okay? Many of the forams that we find are fossils. The White Cliffs of Dover are known for all the foraminifera or forams that they have contained. So it's part of that limestone. Amoebozoa, different than Cercozoa. Cercozoa has the amoeboid nastiness with that hard shell or test. The amoebas don't have that hard shell or test. The amoebozoa include the amoebas as well as slime molds. So when we went earlier, we talked about water molds, different than a slime mold. So you can't just put all molds together. I'm just pointing that out because I want to help whenever, if you re-listen to this or if you're studying and you see mold, I don't want you to put all molds together. So we'll talk about each. For amoebozoa. The amoebas, we have a model and a slide set up in lab, but the amoebozoas, zoa tells you it's animal-like, is a type of protist, single-celled, that have pseudopods, extensions, that they use for cytoplasmic streaming. So basically what that means is they kind of push that pseudopod in a direction, and then they pull the body with it. I discussed this type of movement in 1406, and I used like the slappy hand that you get at like the pizza places and the 25-cent machine, that when you first get that sticky slappy hand and you throw it and it sticks, it's hard to kind of peel it away. So what the whole reason, well, as a parent it disgusts you, but as a kid you don't think about it. As a kid you get it and you let it stick and you think it's really cool. But as a parent, when it sticks to something and it comes back and it has little pieces in it, what does that make you think? Like it's picked up dirt and nastiness. But that's what an amoeba does. It throws itself towards whatever it is that it's trying to prey on, and it kind of pulls itself towards it so that it can engulf it. It doesn't have a mouth, okay, and I'm going to kind of show you. Here are those characteristics. You can find them in environments, marine, freshwater, soil. It talks about their pseudopods, that they have no specific body shape. That's that blobbiness or globbiness I was referencing. Heterotrophic may cause amoebic dysentery. That's just one example. But here, and this animation kind of shows you that how it goes after its food. So here's an amoeba, which is a protist, and look at what it's preying on. Bacteria. Which one of these is prokaryotic? The bacteria. Which one of these is eukaryotic? The amoeba, which is a protist, and protist is eukaryotic. Nonetheless, it has sighted in its food. And so this small bacteria, it kind of throws its nastiness around, and it begins to engulf it, and it will digest it. It moves pretty slow, but it's a really cool process. So if you don't watch this animation, maybe watch one on YouTube. There's plenty of them on YouTube. Slime molds. Slime molds can be single-celled, which is what we call cellular slime molds, or they can be plasmodial slime molds, which is like a multicellular. Okay? So different types of slime molds you can kind of see here depicted. Kind of looks slimy. Like visually you can see that. Well, besides the fact that where they grow, we call it slime molds, you can use them in molecular research. When I say molecular research, what am I studying? DNA. We're studying genetic material. Okay? Some of them have multiple nuclei, so they're multicellular with multinuclei within one cell. So it's a pretty unique setup. But slime molds, these again fall under the amoebozoa. So the amoebozoa includes amoebas and slime molds, but not water molds. Archaeoplastidia includes the kingdom Plantae and two protist groups. All of them are photosynthesizers. Now, we are just talking about the protist part of this. The two protists that it contains are red algae and green algae. Red algae is rhodophyta, green algae is chlorophyta. Both of them carry out photosynthesis. Guess what red algae is named after? The fact that it's red. It mentions here it has chlorophyll pigments, but it also has a red pigment called phycoerythrin. Erythrin is a reddish color, like our red blood cells are called erythrocytes. So that gives you an indication that we're talking red. Again, it's a marine organism. You have used it. It's a component of the agar that we use, our agarose gel. It's also used as carrageenan to give thickness to puddings and ice creams. So you find a lot of protists in your foods, or at least they're used, not specifically the whole protist, but still. So red algae, and there's an animation that goes along with that. Chlorophyta is our green algae, clearly called chlorophyta because it's green, has chlorophyll. It does have cellulose in the cell wall. However, it's unicellular, and all plants are multicellular. So it itself cannot be considered a plant. It tried to be a plant and it failed, just short of becoming a plant. Your volvox that you looked at, hopefully last week, that's an example of chlorophyta. So here's volvox right here. Also in the lab, there's on that back table, there's a setup of lichens. And does anybody know what lichen is? It's a type of green algae. It's a symbiont. And a lichen will create a relationship. It's usually, if you've studied primary and secondary succession, like if it's just bare ground, the very first thing to develop will be a lichen. So these lichens start to break down the rock and over thousands and millions of years create soil, and then that soil will lead to plants and so on. So lichen are a protist, a type of photosynthesizer that can live on rocky terrain. But we also see it a lot on our trees. And so there's also tree branches in lab that show lichen on it as well. It is a symbiotic relationship. It does provide food for whatever it's on. However, if it's growing on a rock, the rock doesn't need the food. But we see like a symbiotic relationship with trees because the tree will give it water and it will help with nutrients. So another indicator lichen brings up, and you always know that you're in kind of like a wetland or there's an area, a creek or water nearby if there's lichen. And usually the lichen grows on the side that's closest to the water. So all of you naked and afraid people who get stranded somewhere, when you identify lichen, you can be like, I know there's water somewhere. Like, I'm getting close. Okay, but for sure check it out in our lab. We obviously believe that this was the last step before plants. So after this, and when we study plants, we'll study bryophytes, and the very first thing it'll say is we believe that these came from the charophytes, from chlorophyta. So we'll get there. Green algae examples also in our lab. Here they believe is the ancestor to plants. This is a great setup. It gives us the shape of a plant, but this right here, even though it's really blown up on this slide, is just a few millimeters tall. These can't be considered plants because they're lacking vascular tissue, which is what you and I will study as xylem and phloem, and that's what gives water and nutrients to a land plant. So if I take this out of water it will not survive because it cannot conduct water from its environment. It has to actually be immersed in water. But this right here is a really, really tiny, tiny plant.
You said that's unicellular?
This one is not.
Okay.
This one is not. So these can also form in colonies. Well, we talked to, it said colonial. Okay. I was like, wait, did I? Because like Volvox is a colonial one. This leads to the charophytes. So chlorophyta is unicellular. Let me go back just to make sure that you understand that. Chlorophyta is unicellular. So it grows and it can single-celled green algae. Then it will grow in itself into colonies. So colonial, single-cellular, that's Volvox. Then the charophytes are where we start to see differentiation. And we spoke on differentiation, didn't we? Like we have a whole colony and some of it takes on specific features or jobs. Okay, I thought we did. So those are the ancestors. Now as we get to multicellular, which we will get to, this right here is lacking that vascular tissue. So it cannot itself be classified as a plant. Good question. Last group of protists is the opisthocons, and the opisthocons are the chlorophytes / charophytes will lead us to plants. The opisthocons will lead us to animals. We believe that the cyanoflagellates are the precursors to our sponges, which we will learn when we get to animals as having cyanocytes. And those cyanocytes look very similar to this single-celled organism. Why can that not be an animal? It's single-celled. It's single-celled. But they will form in a colony and appear to have a multicellular organism. However, individually, they are just single cells. So the cyanoflagellates are the ancestors for, it mentions fungi and animals, and we will study fungi, of course, on Wednesday, but animals later on in this semester. So you'll hear us come back to each of those. So opisthocons lead us to animals, and the chlorophytes / charophytes will lead us into plants. And that is the end of protists. Questions? Okay.